Determining the matrix polysaccharide composition and crystalline cellulose content of plant cell walls starts with dried plant material, which is subjected to various extractions. The resulting material consists of cell walls only and then is split up. To determine the matrix polysaccharide composition, the wall material is treated with a weak acid to hydrolyze non-crystalline polysaccharides, which are mainly the hemocelluloses, into their monosaccharides. The monosaccharides are then derivatized into their corresponding volatile alditol acetates, which are then analyzed and quantified by gas chromatography coupled to a mass spectrometer. For the determination of crystalline cellulose content, walls are treated with the Updegraff reagent, a mixture of acids that strip away non-crystalline wall components. The remaining crystalline cellulose is degraded into its monomer glucose, which can be assayed colorimetrically. The determination of lignin content and composition is demonstrated in another JoVE video publication entitled Comprehensive Compositional Analysis of Plant Cell Walls, Part 1, Lignin.
Hi, I'm Markus Pauly, head of the Wall Analytical Facility at the Great Lakes Bioenergy Research Center here at Michigan State University.
I'm Cliff Foster, manager of the Cell Wall Analytical Facility. Today we'll show you how to determine the composition of the polysaccharides present in the plant cell wall material, that is cellulose and various hemocelluloses.
Here at the Great Lakes Bioenergy Research Center, we develop technology to convert these sugars into biofuels. In order to identify an optimal plant feedstock, we first need to assess the kind and abundance of sugars that is present in this biomass.
In another video entitled